now. And Cam, you can go whenever.
Alrighty.
God, we've already gone 5 minutes over.
Okay, okay. All right, so yes, hi everyone. My name's Cameron, and I'm a new postdoc, as Dan said, in the Hurley lab at the University of Washington. And what I'm going to talk to you, so I've just started here this year. So what I'm going to talk to you today is actually work that I did during my PhD, which was back in Australia at the University of Adelaide. And this was in the lab of Associate Professor Dan Peet. Dan is predominantly a hypoxia signaling researcher. And this project was in collaboration with Professor Bob Casson, who brought the retina expertise. And the idea of the project initially, before I got there, was to assess the role of the hypoxia-inducible factors in retinal metabolism. And then it sort of morphed into a project that was designed to just assess what drives the lactate production in the retina. So in the first half of my talk, I'm going to go through some of the data that I generated trying to answer that question of what drives the lactate production. And then in the second half of the talk, I'm going to jump out of my own data and into the literature to try and explore some ideas about why the retina is even producing so much lactate in the first place, or why any cell type for that matter would engage in aerobic glycolysis. I'm not sure there's a good answer out there, and some of you are probably will know everything that I'm going to talk about, but maybe it's useful to hear someone else think things through. All right, so how can I get rid of this thing at the top of my screen? All right, anyway, I can't see the titles on my talk. But anyway, so oh yeah, by the way, if you like watching in full screen, you should be able to put me up in the top right corner and I won't be blocking any of the info in the slides. So the retina as a whole exports a huge amount of lactate from the glucose that it consumes, something like 79 to 94%, I think, depending on the species and depending on the paper that you read. And this is a very inefficient form of generating ATP compared to using pyruvate to fuel the Krebs cycle and oxidative phosphorylation. This is an old Winkler paper, and in this paper he's used retinas that have been perfused with a medium that is oxygenated with 95% oxygen. And even under this condition, the retina exports about 90% of the glucose that it consumes as lactate. So it's very unlikely that this is due to a lack of oxygen. It's also true that this happens with the neural retina and eye cup incubated intact together, and also when it's measured in vivo using anesthetized animals with measuring arteriovenous differences in the metabolite concentrations. Okay. So a long time ago now, I guess, it was thought that the Mueller cells were sort of mainly responsible for the lactate production. And although it's no longer really thought, it's now thought that the photoreceptors are mostly responsible. It's sort of understandable why this was the case, because certainly Mueller cells, when you put them in culture, they do exhibit a really strong aerobic glycolysis. 99% of the glucose that they consume, they export as lactate, which is higher than the retina itself. And it's not only like long-term cultured Mueller cells that do this, also like acutely isolated Mueller cells will export more lactate into the medium. This is an acutely isolated guinea pig Mueller cell will export more lactate into the medium alone than in complex with photoreceptors. So clearly the Mueller cells rapidly adapt their metabolism when they're taken out of the whole tissue, and this particular result I can't really explain. It doesn't fit with the current understanding, I guess. But the reason that I'm talking about this is because the first part of my talk is using some Mueller cells as a model of retinal metabolism, specifically the RMC1 immortalized Mueller cell line. I'll go over it quickly because it might be interesting from a signaling sort of standpoint, and then I'll quickly get into like whole retinal data after that. The approach that we sort of took for this was to look in the cancer literature, because many cancer cells obviously also exhibit this high lactate production, and there's a lot known about what drives aerobic glycolysis in cancer. And so we borrowed on that to see if the same signaling pathways were involved in the retina. One of the most common in cancer is PI3K signaling, where you get activation of PI3K and then activation of the canonical effector AKT, and then various downstream effects that can upregulate glycolysis. So I treated the RMC1 cells for 2 hours with inhibitors, and then I changed the medium and inhibited for a further 6 hours, collected medium for a lactate assay. And in these RMC1 cells, PI3 kinase drove about 50% of the lactate production. Over here, you can just see that with the PI3K inhibitor, I'm losing phosphorylated AKT, so it's working. Also, when I inhibit AKT, the inhibitor clearly works. It gets rid of the phospho-AKT. However, the AKT inhibitor had no effect at all on lactate production. And this is a kind of unique result as far as I've been able to tell from the literature. I haven't seen another cell type where PI3K is driving glycolysis quite so independently of AKT. There was also a small effect when I inhibited MEK, but I didn't really follow this one up. I also used a different PI3K inhibitor just to make sure that this was a real effect, and I saw a similar difference. So PI3K is definitely driving the lactate production in these RMC1 cells. I then went back to the literature to see what else downstream of PI3K might be driving glycolysis other than AKT. And there's really only this one paper that shows that RAC1, which is a small GTPase, and it results in cytoskeletal remodeling and release of aldolase from the cytoskeleton, and that can drive glycolysis. So I used this RAC1 inhibitor, and it did indeed seem to be involved. So I saw this decrease in lactate production, but it wasn't a super clean result because it also decreased the protein content in the cells. So when I normalized the protein, like I was showing with those previous results, I saw no significant change, which was a bit weird because the total number of cells was the same. So I'm not entirely sure what this is saying, but basically the conclusion with the RMC1 cells is that the PI3 kinase drives aerobic glycolysis completely independently of AKT, and it's possibly through this RAC1 pathway. So Mueller cells are probably not the best model of whole retinal metabolism. That's becoming sort of increasingly clear, and I think it's pretty solid now. So for example, it's Mueller cells in vivo don't seem to express any isoform of pyruvate kinase. This is a mouse retinal staining from this Lindsay paper, where pyruvate kinase M2 is expressed most highly in the photoreceptors and basically not in the Mueller cells. Similar with PKM1, expressed highly in the photoreceptors here and also through the inner retina. And admittedly, in this particular image, it's hard to tell, but in the paper where they've looked closer, there's not any overlap between PKM1 and the Mueller cells. Also, this fluorescent analog of glucose 2-NBDG is taken up more highly into the photoreceptors than into the Mueller cells, shown here in the mouse retina and also in the zebrafish retina. I then wanted to look a bit more closely, I guess, at the sort of developmental differences in metabolism between the Mueller cells and the photoreceptors. So what I did was I looked in the literature for RNA-seq data sets over the development or sort of the postnatal maturation of the photoreceptors as they grow their outer segments and mature metabolically over about 4 weeks postnatally for the mouse. So I found these 2 studies. There's this Swarup group paper where they've isolated a GFP tagged NRL, so NRL promoter-driven GFP. FACS isolated these photoreceptor cells from P2 to P28. And over here, for this this other study, they've looked at Mueller cells. So either SOX2 GFP mice for retinal progenitor cells at P2, or for the later time points, RLBP tomato mice. And I've analyzed these data for expression of genes involved in glycolysis over this postnatal development period. So it's a bit small, but this is P2 on the left up to P28 on the right, and P2 up to adult on the right here for the Mueller cells. And sort of, I guess the main takeaways from this that I got were that in the photoreceptors, the important genes for aerobic glycolysis, so for example, hexokinase 2, also hexokinase 1 down here, PKM, aldolase, LDHA, they're all upregulated, most of them by sort of 10-fold, hexokinase 2 by 170-fold is huge. Whereas in the Mueller cells, some of the genes involved in glycolysis are upregulated, hexokinase 1, 1.7-fold, some of these others, aldolase, but there's also important ones that are downregulated, like hexokinase 2 and LDHA, although their expression is still quite like reasonably high, I guess. Aldolase here is a bit of a weird one that gets massively upregulated, and I'm not sure what that's about. So basically, the conclusion here is that the rods are certainly the more the glycolysis is driven a lot more in the rods over this developmental period rather than the Mueller cells. And just to illustrate this, at least one isoform of every step of glycolysis is upregulated in the rods over that developmental period. So we changed model to these rat retinal explants. And in these experiments, I was scavenging tissues from Sprague-Dawley rats. So in each individual experiment, it was either all males or all females. The male rats, when I used them, were 8 weeks old. The females were 6 weeks old. And once I'd normalized the protein, there was no difference at all. So what I'm showing you today is just lumped all together. So what I've done is the rats were euthanized by CO2. I dissected the retinas, I put them in a dish, and then I started treatment with these inhibitors, the same ones that I showed before with the Mueller cells. Incubated for 5 hours, then changed the medium and incubated for a further 11 hours, and then collected the lactate, collected the medium for a lactate assay. And I used this time point. Some of you might think it's a bit extended because I wanted, someone raised their hand. Did you have a question? You can go if you have a question. Otherwise, I'll keep going. Okay, I'll keep going. So, yeah, so basically I used this longer time point because I wanted to make sure that the inhibitor had time to really enter the cells that it had to, and also for any downstream effects to take place. So turnover of protein, transcription, translation, because I don't know what mechanism is going to be involved. So firstly here, I've just looked at the glucose consumption and the lactate production over this period. And if you crank the math on this, it works out to about 92% of the glucose consumed being converted to lactate, which is very much in line with the past literature. And also inhibiting LDH basically abolishes the lactate production. So it's being produced in the sort of way that you'd expect. All right, so I then used the same inhibitors as before, but I saw no change with the PI3 kinase with 2 different inhibitors at 2 different concentrations. Although there's maybe a slight trend. The problem with this was, so I don't have Western blots for this because I wasn't actually able to detect any phosphorylated AKT in the retinal samples, which is just, in my mind, further confirmation that this pathway is probably not constitutively driving the lactate production. And as you can see here, also with the AKT inhibitor and the RAC inhibitor and the MEK inhibitor, I saw no change. I then sort of started looking a bit more broadly in the cancer literature and tried a couple of other things. 2 different mTOR inhibitors had no effect on the lactate production, and also a Wnt signaling inhibitor that inhibits tankerase had no effect.
Hey, Cam.
Yes.
the medium that you used to culture these retinas in, was it the same as the medium you used to culture the Mueller cells in?
It was so the medium that I used for the Mueller cells was a DMEM with 5.5 millimolar glucose. This was MEM with 5.5 millimolar glucose.
Sometimes these media have like additives that could be stimulating signaling pathways, but if you're just using
Yeah,
a
so I tested, I haven't got it here. I tested including FBS, 10% FBS or not, and I saw no difference in the rate of lactate production when I included it. So I left it out for all of these experiments to avoid sort of unnecessarily stimulating pathways if it wasn't affecting the rate of lactate production. Yeah.
Cool. Thank you.
So there was one mouse paper that showed that fibroblast growth factor receptor drives lactate production in the mouse retina. So I've used the same inhibitor at a couple of the same concentrations that were used, and I saw no effect when I treated these rat retinas under the conditions that I just said. So I tried to match the conditions a bit more closely by using neurobasal A media with 95% oxygen environment. So these previous experiments, I should say, were done under just room air. So when I did this, I saw a decrease in lactate production by just having the oxygen there, which is not too unexpected. And then there was this small but significant decrease in lactate production with the FGFR inhibitor at quite a high concentration. Although it's a similar magnitude effect here, so it might just be the fact that this was slightly more consistent in terms of the error bars. I was a little concerned that this inhibitor was quite a high concentration from my reading, so I wanted to investigate this a little bit more. The idea in this mouse paper was that FGF2 released by the RPE activates FGFR in the photoreceptors to drive lactate production. So what I've done is I incubated either retina and eye cups separately, or I kept them together intact and incubated them and then split them up just before the Western blot. And in either case, I saw phospho-FRS2 and phospho-ERK, which are indicative of active FGFR signaling, only in the eye cup and not the retina, which is sort of the opposite to what I was expecting. And even when I treated with human FGF2, I couldn't seem to actually activate FGFR signaling in the retina. So in my mind, I think this in the rat retina is probably an off-target effect. This might be like a difference between the rat and the mouse. I'm not entirely sure, but I basically didn't find the same evidence that FGFR was driving lactate production in the rat retina. I then took a totally different approach to this whole question. Instead of looking in the cancer literature for ideas, I went back to this developing photoreceptor RNA-seq data set, and I asked, what if there's a transcriptional driver that has a similar pattern of expression to a lot of these glycolytic genes that might be involved in driving their expression. So I got a list of transcription factors. You can see most of them are downregulated over this developmental period, and there's a small subset that are upregulated. And over here, I've just organized them by fold change. And then I went through one by one and looked in the literature to see which would potentially be involved in driving aerobic glycolysis. There's a couple that might be interesting, but the first one here, estrogen-related receptor beta, which is upregulated 1800-fold, is probably the prime candidate. And that has been shown in the past. There's a study that knocked it out and showed retinal degeneration in the photoreceptors, and also decreased expression of some metabolic genes in glycolysis and in oxidative phosphorylation, but they didn't show change in the lactate flux. So I really wanted to test that at the level of the actual lactate. So I got a hold of a couple of inhibitors of this estrogen-related receptor beta, diethylstilbestrol and 4-hydroxytamoxifen. And when I treated with the DES, it did knock off a portion of the lactate production, but there was no significant change with the tamoxifen, which to me was a little odd because they should be doing the same thing. So again, I started looking a little deeper. I ran a dual luciferase reporter assay where I had 5-fly luciferase driven by the estrogen-related response element. When I transfected in the estrogen-related receptor, I got an increase in firefly, as you'd expect. And then treating with either DES in the pink or 4-HT in the green, I saw a drop-off with increasing inhibitor concentration, which is also what you would expect. However, the constitutive ranilla was also dropping off at the same rate. So basically, I'm just getting general transcription inhibition, or I'm killing the cells essentially. And that was quite clear that cells were dying. So I'm not entirely sure if, basically, I can't confirm that this result is specifically due to inhibiting estrogen-related receptor beta, or if it's just death of cells. And I should say that I didn't only do this in RMC1s, I did it in 293Ts as well, just to make sure that it wasn't a cell type specific effect. And I saw exactly the same thing. So basically, I think this question can only be answered by like a genetic targeting approach. And I think that might have been done, but I'm not entirely sure.
Cameron,
yeah,
it's really interesting data. Did you look at the cancer cells?
Sorry, hang on, I'll put in my headphones.
Okay, maybe I'm too quiet. Have you looked at cancer cells, breast cancer cells especially?
No, I didn't for this. I haven't looked at those, no. Yep.
All right. Yeah, because this drug, I mean, indeed, it's not good for many because it actually goes to every nuclear receptor.
Okay.
It binds it, especially in a high concentration. So you might want to go to some cells which have more expression, really more expression of this one.
Okay.
To others. Okay, just a suggestion.
Yeah, thanks. Okay, so yeah, this sort of includes my my personal data that I generated. And so what drives lactate production in the retina? I don't know. But maybe in this second part of the talk, I can get us closer to an answer, or maybe closer to just better ways of thinking about it than what I was doing here. So for what reason does the retina, or any cell type for that matter, actually use aerobic glycolysis? I think it's a really interesting question, given that it's such an inefficient means of metabolism. So I've looked at the cancer literature and the retinal literature, and I've come up with a few sort of domains that the reason might fit into. So firstly, maybe the lactate is used as a fuel for other cells. Maybe it's for the primary purpose of ATP production, or maybe it's for biosynthesis or redox homeostasis. So I'll go through these one at a time. Firstly, maybe it's used maybe the lactate is used as a fuel for other cells. I think this can happen. I think other cells can take up some of the lactate and use it. So for example, here, this paper is using RPE cells, and they've shown that certainly the cells can take up lactate and put it through the Krebs cycle. However, with any experiment that fits in this sort of, with any hypothesis that sort of fits in this domain of the lactate being a fuel, it doesn't get around the fact that 90% of it is exported. So why wouldn't the cells just use the glucose to fuel? Like, it seems like such a waste of the lactate. So in my mind, this can't be the primary purpose, even though it may happen. In the second sort of domain is ATP production. So this seems initially counterintuitive, but there are a few ways that this might be the case. So ATP is produced more rapidly by glycolysis, I'm sure most of you know, than by oxidative phosphorylation, even though it's so inefficient. So this led to the idea that rapid fluctuations in ATP demand might drive glycolysis. So this as a model looks something like this, where the base load demand of ATP production is met by oxidative phosphorylation. And then if you have rapid spikes in demand, it's met by glycolysis. And this is some data from the cancer literature that sort of shows evidence for this. So in this paper, they've looked at a bunch of different cancer cells, and in their evidence, the sodium potassium ATPase is a rapidly fluctuating demand, which I'm not so sure of, but that's what they've said. So here they've shown that activation of the sodium potassium ATPase with gramicidin increases the proton production rate and decreases the oxygen consumption rate for all of these cells. And then similarly, when you inhibit the sodium potassium ATPase, there's a big drop in the proton production rate and almost no change in the oxygen consumption rate. This is an interesting idea in the retinal context, because even certainly the retina probably has quite a fluctuating demand. Even as you look around the room, the rods in your eye are getting exposed to big differences in like light intensity, and that is going to affect the amount of ATP that they're using. So it's an attractive idea from that perspective, but I just don't think it quite matches this cleanly with the evidence that's out there. So for example, when the rabbit retina is treated with strophantidine to inhibit the sodium potassium ATPase, you don't see this clean separation of these lines here. So instead, you do get a large decrease in the lactate production of 58%, but you also see a decrease in the oxygen consumption of about 50%. And this means that the change in ATP production is probably mostly being met by a decrease in oxygen consumption rather than glycolysis. Also the rabbit retina here, when it's kept in darkness and then exposed to flashing light, there is a small increase of 27%. Well, that's a reasonable size increase of 27% in the rate of lactate production. Similarly, when it's kept in light and exposed to dark flashes, there's an increase in the lactate production. However, so you might say that, yes, this is evidence that the retina responds to fluctuating demand with increased lactate production, and maybe it does, but you still have to explain that 75% of the lactate production is already there when the retina's in just steady darkness, or it's already there when it's in steady light. And I don't think that's quite explained. So this is an interesting idea, but in my mind, it's not the one that best fits with sort of the data.
Hey, Cam.
Yes.
Just real quick. I can't remember the exact numbers, but photoreceptors use like 1 to 2 orders of magnitude more ATP in the dark compared to the light, because they're pumping so many more ions.
Yeah.
So what I would expect, if lactate production is driving like ATP synthesis, I would expect a drop in lactate production in the light, because they don't need as much ATP.
I agree completely.
But that data was showing like the opposite, right?
Yeah. Yeah. True. So I guess that's just another reason sort of against that idea. So another reason could be this molecular crowding idea, which is based off of like mathematical modeling. So in this, the idea is that there's a finite volume of the cell that can be dedicated to ATP producing processes. And as you increase glucose uptake from 0, the cell will gradually fill with mitochondria because it's the more efficient method of ATP production. But once it reaches this threshold glucose uptake rate, it'll switch to start using aerobic glycolysis and producing lactate. And this is because the glycolytic machinery can produce more ATP per unit volume than the mitochondria, despite the fact that it's less efficient. So above this threshold glucose uptake rate, you start to see a very small, slow increase in ATP production, which is only sort of valid if you have an extremely high glucose uptake rate. So I think this idea is interesting because the retina of all tissues is probably likely to be subject to this sort of crowding, because the rods are so narrow, I guess, and tightly packed. And if they've got this sort of morphology, it's likely to decrease their cytoplasmic volume. So maybe they reach this sort of species-dependent sweet spot where they're maximizing ATP production. They're consuming a huge amount of glucose and a huge amount of oxygen. And maybe it's just that they're afforded this ability to be really inefficient because having tightly packed photoreceptors is really beneficial to the organism. And yeah, I can't fully rule this out, but again, it's not my favorite idea. My favorite idea fits into this third domain of biosynthesis and redox homeostasis. So there's some evidence that in the retina, the lactate is produced, or aerobic glycolysis is for the purpose of biosynthesis. So this here, glycolytic reliance promotes anabolism in photoreceptors, classic paper. So when mouse photoreceptors are electroporated with shRNA targeting LDHA, you see this large decrease in rod outer segment length. And this is dependent on the catalytic activity of LDHA. So it really suggests, I guess, that the lactate is produced. So when you inhibit the lactate production, you get a defect in anabolic activity. Although I guess an argument against it is that there could be other reasons that the outer segments aren't as long. But so I'll just sort of assume for now that the lactate production is for biosynthesis. I'm not sure about the authors of this particular paper, but I think when a lot of people think about aerobic glycolysis and biosynthesis, they think of the now, I guess, old idea that it's predominantly because the glycolytic intermediates are being siphoned directly into macromolecules. But I think that idea is probably a little. I think there's a better idea out there in the literature now. So this is from a cancer study where they've looked at a bunch of different proliferating cell lines, and I think most, if not all of these, exhibit aerobic glycolysis. And when they've stably labeled them with amino acids or glutamine or glucose, only so the majority of the cell mass the majority of the carbon mass is derived from amino acids. So small amounts of amino acids that are present in the medium rather than glucose, despite the fact that they are exhibiting aerobic glycolysis. So So basically it's showing that aerobic glycolysis is probably not primarily to increase the pools of glycolytic intermediates for their direct siphoning into the macromolecules. It's for it helps biosynthesis in some other way. And I think this paper here goes a long way to explaining it, although not all the way, but it does go a long way. I quite like this one. So in this paper, what they've done is they've inhibited pyruvate dehydrogenase kinase. And when you do this, you relieve inhibition of the pyruvate dehydrogenase complex. So pyruvate can enter the TCA cycle in mitochondria more freely and generate more NADH. And it also will result in decreased lactate production. So you get decreased NADH to NAD+. And that's shown over here. So using this inhibitor, they see decreased lactate production. Also, they see decreased proliferation rate of these cell types. So it's linked with the ability of the cells to proliferate. I guess then also, as you would expect with this inhibitor, because you're getting a higher production of NADH and decreased removal of NADH in the cytoplasm, you see a decreased NAD+ to NADH ratio. So you've got more of this hanging about. And this is rescued by addition of pyruvate. So when you add a bunch of pyruvate to the cell can convert NADH back to NAD+, and it restores this ratio, and it also restores the doubling rate of the cells. So when you when you use another method to remove the NADH, in this case, they've treated with duroquinone, the cell can convert this back to, or can convert it to durohydroquinone, and that will oxidize NADH. NADH. That also rescues the proliferation rate. And then another way of testing this. So the main way that the cell can oxidize NADH is through the electron transport chain complex 1. So if you want to increase the cell's ability to oxidize NADH via this method, one way to do that is to uncouple the proton gradient with an uncoupler such as FCCP. And in theory, this would allow the cell to run this complex 1 reaction more rapidly because it doesn't build up the membrane potential in quite the same way. So when they've treated with this uncoupler FCCP, they see that this does indeed restore the NAD plus to NADH ratio here, and it restores the proliferation rate. So basically, the cell is making too much NADH than for what it can deal with in order for its biosynthetic activity. And these authors then thought that maybe ATP hydrolysis is limiting the rate at which the cell can run this oxidation of NADH at the electron transport chain complex 1. And although these effects are small, there are small increases in the proliferation rate when they treat with this gramoside D, which just increases ATP hydrolysis in the cell. I think there's good evidence that this idea that the cell is in a state where it's making more NADH than it can comfortably deal with is probably true in the retina. So for example, and I think the retina is already taking steps to deal with it. So for example, the photoreceptor mitochondria are highly uncoupled. When you treat a retina with oligomycin to inhibit ATP synthase, only 50% of the oxygen consumption is lost. So this other 50% here is basically oxygen consumption that's uncoupled from ATP production. And I think there's even weirder stuff going on that shows that the retina is taking steps to try and increase its rate of NADH oxidation. So for example, again here showing this loss of 50% with oligomycin. If you then add marlinate at increasing concentrations, which inhibits this complex 2 of the electron transport chain, you see no further decrease in oxygen consumption at all. And this to me suggests that basically the cells aren't depositing electrons here at complex 2. And then when you treat with rotenone, which inhibits complex 1, you see that the rest of the oxygen consumption is completely lost. So certainly the cells are depositing electrons here to remove the NADH. And there's some interesting experiments out there that show that under hypoxia, the cells can run this complex 2 reaction actually in the opposite direction. And in this case, when this happens, fumarate kind of acts a bit like oxygen and becomes a terminal electron acceptor in the electron transport chain. Electrons can be deposited at complex 1 and then flow to complex 2 in the reverse direction. And there is some evidence that the retina actually does this. So for one, the retina exports a large amount of succinate. Also, I love this experiment. In this experiment, they've tagged the 4th carbon of glucose with deuterium. So when it goes through glycolysis, the deuterium is transferred to NADH at the GAP-DH reaction. The deuterium can then be transferred into the mitochondria via malate through the malate aspartate shuttle. And if this reaction is operating in reverse, then you would expect the deuterium to label fumarate and then succinate. Whereas if it's going in the forward direction here, you lose it to NADH. And indeed, in the retina, these are retinas in a dish that are incubated in normal oxygen, in normal air medium. You do see this labeling of malate and fumarate and succinate, which suggests that at least in room air, in a retina, this reaction is operating in the reverse direction. And in my mind, this could be the cell sort of screaming out for additional electron acceptors. And maybe this allows it to oxidize NADH at a faster rate than it can do with oxygen as the only terminal electron acceptor. So I think the retina is really in this state where it's making a lot more NADH than it wants to. And aerobic glycolysis is basically the cell's way of removing NADH. So that's sort of obvious when you think about it from the fact that the LDH reaction is in really rapid equilibrium. So if you're getting a bunch of lactate production, then obviously you're getting a huge amount of NADH production. And I think the NADH is generated from biosynthetic reactions, rather than this being a use of the glucose carbon for macromolecules. So basically, to finish off, I've got this kind of stupid analogy. So basically, you've got this pond, and in this pond, there are a bunch of frogs, and they're making tadpoles. And usually, the tadpoles will flow off this waterfall, never to be seen again. So then, if the frogs start making, so sometimes the frogs get a bit too excited and they start making even more tadpoles. So there's this guy, LDH, who stands at the top of the waterfall with his buckets, and he scoops out the tadpoles and he puts them on this conveyor belt, never to be seen again. So he's happily doing this, but the frogs, they start making too many tadpoles. So he's running out of buckets. So he calls out he needs more buckets. So glycolysis is here. He's got buckets, but they've already got tadpoles in them. So he doesn't care. He just pours out his tadpoles into the pond and then gives the buckets to LDH. So basically, I think this is what's going on. I think the cell, it needs pyruvate in its cytoplasm so it can remove excess NADH. But this picture isn't really complete because it doesn't explain why the cell would increase its glucose uptake, I guess. So there are a few unanswered questions here. So what are the frogs doing exactly? Like, what is the source of this NADH? Specifically, it's probably, in my mind, biosynthesis. But what are the specific reactions that the retina is undertaking that creates all this NADH that it can't deal with? Is glycolysis really this useless? Or is there something that we don't understand about how it affects the redox state of the cell that means it's a more effective way of eliminating NADH than it sort of appears in this picture? Is ATP hydrolysis limiting the rate of NADH regeneration in the retina? So is it sort of blocking this waterfall? And then how can we help? So I think one way is that we can provide more buckets, that being more pyruvate. So I think if you have a cell and you think it might be under sort of metabolic stress of this type, I think it would be potentially useful to try treating with pyruvate and seeing what it does. We can sort of provide other ways of removing NADH from the cell. And maybe we can try and calm down the frogs. So if we work out what's generating all this NADH, maybe the frogs think it's really important what they're doing, but maybe they're doing more damage than good. Maybe if you calm it down, the photoreceptors will still sort of survive for longer. And maybe we can clear the path of this waterfall if it turns out that ATP hydrolysis is a limiting factor here. Maybe we can add sort of futile means of fixing this. So I had a couple of just weird ideas to finish off with. So first, if you think about it, if ATP hydrolysis is the rate of ATP hydrolysis is a problem here, then you'd think that metabolic problems would be worse in the light where ATP hydrolysis is significantly lower. And I think this is true. So in light-induced photoreceptor degeneration, for example, you would think. So I've looked actually, and there are a few papers, although not widely seen, I think, where pyruvate is actually shown to protect against light-induced photoreceptor degeneration, which would fit with this idea that there's a problem of overproduction of NADH and a limiting factor being the ATP hydrolysis rate. And then I had another thought, which is a bit weird, but I'm not sure. Does anyone know if the actual fundamental reason why the photoreceptors are these weird backward neurons that are constantly constantly in this partially depolarized state with the dark current? Because if that's not fully explained, then maybe it's like a mechanism that's evolved specifically because it's a kind of futile use of ATP. Maybe it benefits the cell to use a ridiculous amount of ATP because it allows them to regenerate NADH faster for whatever else they're doing. Right. So, yeah, in conclusion, I'd just like to thank all these people at the University of Adelaide and in my current lab at the University of Washington. Yeah, thank you all. Happy to take any questions.
Oh, sorry, Dan. Oh, you got a.
Sorry, could you start again? I just plug in my headphones.
That's okay. Cam, this is Jason Miller. I'm at University of Michigan. I mean, this is you're in the realm of total speculation, which is great for these talks. If you're going to totally speculate, about why the dark current exists and whether or not it's to just, dissipate this reductive stress, why not postulate better expression of uncoupling proteins, in the mitochondria instead of the dark current? There's no right answer to that, but I'm curious.
I certainly agree. That was just a weird thought that I had last night, and I thought I'd throw it in at the end of the talk. But you're right, there's probably other ways to use up. I mean, so the retina, it's already highly uncoupled. Maybe it's at a point where it can't uncouple anymore for some reason. I'm not sure.
Thanks again. That was really interesting. I agree with Jason. It's cool to like throw around these ideas and try to figure out what's going on. My understanding of glycolysis, and it could be wrong, is that you get a net production 0 NADH from turning glucose into lactate.
Yeah.
Is that correct, or am I?
That's correct as far as I understand.
So then how are you dissipating excess NADH with aerobic glycolysis?
No, so that I don't know, basically. So that's what I mean by that. That paper doesn't have a complete explanation for it, for aerobic glycolysis. I think I think there's good evidence that the cell is in a state where that's what it's trying to do. I just don't know if it's understood whether upregulating glucose uptake and running glycolysis at a fast rate actually helps the cell or not. It seems to, but I don't know why. Yeah, and I don't know if anyone knows why yet.
This is Jen. Hi. Cameron.
Hi.
Okay.
Hi. Yep.
I think you did a great job, to organize all this stuff and your data and also the literatures. You did an excellent job and also
Thanks.
really creative to make those tadpoles. I really enjoy your talk. Yeah, really, really nice work. So I want to add a couple of things. I think the NADH is a very, very, very interesting ideas. Some of them maybe fit to that hypothesis. When we do some study, also what we learned from this, so in the retina, if incubated, they use a lot of aspartate. So probably they can add more oxalic acetate, then help to regenerate the NAD. I'm curious, if you add aspartate, maybe lactate release can be a little bit different, maybe a little bit different in vivo and in vitro.
Okay.
Also in the cones, if you look at the cones, they're supposed to use more energy, but the cones use more pyruvate than the rods. If you compare the cone-dominated and rod-dominated tissues, so maybe the pyruvate in the cones, there's a higher need of this, remove this NADH to NADs.
Okay.
So they may use more pyruvate. We do not validate that, but from our results shows that cone-dominated retinas use more of the pyruvate. Also, the expression of the genes related to pyruvate utilization is more. So it's very interesting. I think probably in the retina, Jim showed a very nice work, and then the oxygen maybe not enough, or they're located in some conditions, or they use tons of oxygen. They saturated the complex 1 to recycle the NADH. So they have to use some other ways. So one way to do it is lactate dehydrogenase. That is really, really efficient ways. But another way is to generate aspartate. So, I think there's some, because culture system not so complicated to make this. And if you have more complicated culture system, probably it will make aspartate. And what we find recently also in retina, they make a lot of prolines. So they do not catabolize proline, but they make a lot of prolines. So proline seems like also they consume a lot of NADH, maybe another way to use NADHs. So it's very, very interesting ideas. Yeah, I really enjoyed the talk.
Thank you. Yeah, no, that's good. Yeah.
One idea I've been noodling with, which I don't know if it makes a lot of sense at all, but has been that the retina wants to have some kind of high NADH-NAD ratio. I mean, not too high, but it wants to have it above a certain level so that it can catalyze some NADH-dependent reaction that's very unfavorable. And I don't know what that reaction could possibly be, but I've been kind of dreaming that maybe there's just something that the retina does that that it makes use of this excessive ratio. But I agree that it's trying to get rid of it because it doesn't want it to be too much, but it is set up to have that high level of NADH already in the first place, so that must exist for some reason.
Yeah.
Well, through a high lactate to pyruvate ratio, that could transmit a high reducing power like to the RPE also. And if you need that for making glutathione, you can be transferring all that reducing power to the RPE. It's because both the lactate and pyruvate, well, they're released at a certain ratio, and they will be taken up into the RPE at that ratio, and that will determine the NADH to the NAD, and probably maybe connected to the NADPH and NADP ratio in the RPE. Maybe that could be another reason for having such a high reducing power.
Early reveal of a conversation that we've had in
many times. Yeah. Okay. Actually, Cam, I also wanted to ask: do we have to add frogs to our animal protocol?
No, I don't think so. No, okay. Not quite.
All right. Just checking.
What about cartoons to the animal protocol?
Yes, they do have to go in.
Francois, what's up?
So, if I may, so I thought this was a really nice talk. I also think that, well, some of it was be a little bit speculative, perhaps, the second half, but since nobody really understands why this lactate production happens, I think you're allowed to speculate a little bit. And maybe you'll get some new ideas out of that. You mentioned these 3 different possibilities that you were envisioning, and I think the speed of ATP production may actually be really interesting. You quoted this one paper where they were talking about cancer metabolism, where oxidative phosphorylation would kind of satisfy the base production of ATP, and then aerobic glycolysis would come in to deal with fluctuations in demand. And neurons, when you have an action potential running, you need ATP now on a millisecond timescale. And the same happens in photoreceptors. You need ATP in an instant, not like a minute later, because then you're dead already. You need it now. And glycolysis may be the mechanism. I'm not sure whether it's the only, but it's certainly one mechanism that can satisfy the speed of production that you need. And especially, as you mentioned, to fuel NKX that is needed to repolarize the cell membrane. And then you mentioned that there were some older studies, I think Ames 1992 or so, where they had used sulfentidine to block NKX, and you said that this was not really consistent with the idea that the ATP production was used to fuel NKX. And I think one should perhaps revisit those old studies, because sulfentidine may not only inhibit NKX, maybe it's something else. Maybe one should use some new tools, genetic tools, for instance, to see what happens if you really block only NKX or knock it out, if that's compatible with life.
Thanks. That's
anyway, just some thoughts. Very nice.
No, certainly. I wasn't aware that sulfentidine had other targets in that. So thanks.
So there's one comment I think Cameron probably missed out for that is lactination. So there's a nice paper by I think I don't pronounce it correct or not, is Guillermo Oliver from Northwestern University. Very nice paper published recently. And they show that lactate production is important for eye morphogenesis. So if they block that, it will not produce the right eye in the fish models. Very nice work. They also do a lot of signaling work and epigenetic work if it blocks the lactate production process.
Kim, can I ask a question? Maybe it was and Francois. So I understand the idea, you know, that you would have some oxphos takes care of baseline ATP and then spikes happen with quick, you know, glycolytic changes. But photoreceptors are still different than all neurons have this axon firing potential issue, you know, on, off, you know, occasionally needed not. And yet something so unique about like the just dramatic amount of glycolytic sort of requirements for photoreceptors compared to any other neuron. So I guess I'm trying to reconcile that principle, you know, with the fact that yes, photoreceptors, just like any neuron, have, you know, quick energetic, you know, need changes. It's just that their need changes are in reverse. They're chronically depolarized and then they, you know, repolarize. So I'm trying to figure out how that would explain, you know, something unique to photoreceptors compared to other neurons.
Just quick comment too, sorry to jump in, but I think neurons in the brain and CNS, they use lactate as their main fuel source derived from glial cells, not glucose either. So they're kind of backwards to photoreceptors in that respect as well for their metabolism.
Yeah,
at least that's the current understanding, which could be like it was with Mueller glia and photoreceptors back before Jim did all of his amazing work showing that ecosystem.
And like to follow on Eric's point, I mean, at least maybe I don't know how much other people think about it this way, but I think of the RPE as, you know, just like a brain glial. I mean, it lost its identity and became an epithelial cell because it's got other things to do, but it's, you know, got many, you know, glia-like functions. And the metabolic relationship between RPE and photoreceptors feels in some ways reversed on the lactate side to the way it is in brain. Yeah.
Well, I think not everybody agrees with the astrocyte neuronal lactate shuttle, and it could depend on where the neurons are, you know, what environment they're in. Sometimes they'll use lactate, sometimes they'll use glucose. And I think another big difference between a neuron in the brain and a neuron in the retina is the retina's, you know, exposed to a lot of light. And that light energy could be used to induce oxidative damage. So there could be reasons to have really low levels of oxygen in the retina. And so, you know, like Francois was saying, we're really in the realm of speculation, but that's another big difference is that it's you know, it's being exposed to a lot of energy coming from light.
Well, this is Jim, what you and Dan have talked about, that like the goal actually is to get the retina down to 0 oxygen so that you don't have to worry about the ROS, right?
That's a hypothesis.
Yeah,
Yeah.
Also, I think I follow the Jason's comments on RPs. What we find is we still do not know, but what we find if one of the cells, RP cells, whatever they have fibrosis, like an EMT process or using different ways, one of the major changes is lactate utilization. They lost if you look at any databases, like TGF-beta reduction or different ways, one of the changes is lactate transport decrease. If we look at the nutrient use, they cannot lose ability to use lactate. Maybe lactate in some ways is really important for the RP differentiation as well.
Yeah, I mean, to continue on that, you know, could be, I mean, there are lactate sensitive GPCRs. It could be like a signal of, hey, the retina is healthy. You know, like maybe all the surrounding cells want to know if the retina is healthy or isn't healthy. And this is, yeah, I mean, like it was super speculation, but if lactate is a signal that the retina metabolism is sort of normal, then a decrease in lactate could be sensed by other cells that they could respond theoretically. Rob.
Hi. So that was really interesting. I'm at Northwestern. I'm I don't really know Oliver, so I guess I have to get to know. So I'm not a biochemist, but a couple of things occur to me. One is with respect to the speed issue. If you look at pH changes in the retina in vivo, they're quite slow. So in the cat outer retina, they take, you know, quite a few seconds to develop. So that argues to me sort of against this rapid change thing. The other thing is that when you're thinking about whole retina, you know, there are a bunch of different cell types, and you're thinking mostly about photoreceptors, and I guess that's probably valid in rat and mouse, where the inner retina doesn't actually do much aerobic glycolysis, I think.
Yeah.
But at least you've still got Mueller cells and photoreceptors there, so it's complicated. And the other thing that I remember hearing sometime long in the past was that maybe the reason that you've got high glycolysis, aerobic glycolysis in photoreceptors is that it's done in the outer nuclear layer where the metabolic machinery is, and all the ATP made by oxidative metabolism is used for the pumping in the inner segment, and it doesn't leave any for the more other metabolic generative processes.
Okay. Yep. That would, yeah. Okay. That makes a lot of sense. Thanks. Yeah. So certainly, I probably should have taken a bit more time to discuss the species specific differences, I guess, in the talk. I showed some rabbit data, and certainly the rabbits are very different in that they don't have the oxygenated inner retina. So yeah, something I should have probably considered a bit more, I guess. Yep.
In regards to the cell type specificity of lactate production, and again, this is kind of limited to mouse, but I don't know, Nancy, are you still here? Nancy Phil, she generates all these really cool models where there might be like a deletion of GLUT1, the main glucose transporter in the retina, or a pathogen which helps the lactate transporters localize to their plasma membranes. And she sees that when you delete the glucose transporter or the lactate or the thing that helps lactate transporters stabilize in photoreceptors, then you really get an incredibly dramatic reduction in lactate release. That it seems to reflect that lactate production is primarily a photoreceptor activity. Yeah, sorry.
All right. I think, yeah, is that all done? Thanks. Yeah, thanks everyone for tuning in and watching and all your questions. Yeah. It's a good discussion.
Thanks, Kim. It was fun to see speculation.
I love a bit of speculation.
Thank you. Very nice work.
Thanks.
See you all in 2
Everyone.
weeks.